Welcome to the Cannabis Risk versus Benefit Learning Module, Section 1, Cannabis Research Overview. Cannabis use in the United States is increasing. According to a 2015 survey from the Journal of the American Medical Association, usage doubled from 4.1% in 2001 to 2002 to 9.5% in 2012 to 2014. In a 2017 survey, 14.6% of the 9,000 adults surveyed reported cannabis use in the past year. This large increase in cannabis usage may be contributed to in part by the legalization of medical and recreational use in multiple states throughout this time period. The same 2017 survey previously mentioned also showed that 81% of U.S. adults believe that cannabis has at least one benefit, and about 91% of U.S. adults believe cannabis has at least one risk. The most common benefits indicated were pain management, treatment of disease, and anxiety or stress relief. The most common risks indicated were legal problems, addiction, and impaired memory. Interestingly, 22% of the U.S. adults surveyed indicated a belief that cannabis is not at all addictive. In addition to the increasing number of U.S. citizens using cannabis, the demographics of cannabis use are also changing. In 2002, youths were more than 3 times as likely to report cannabis use than adults. However, in 2011, the demographics had flipped, and adults were 2.5 times more likely to report cannabis use than youths. Despite the growing prevalence of cannabis use by adults in the United States for both medical and recreational purposes, there is a dearth of information regarding its use, which leads to 2 main questions. First, why do we not have more data on cannabis? And second, what data is currently available and how can it be interpreted to assess cannabis risk versus benefit? There are several barriers to the research of cannabis in the United States. The first is that cannabis used for research can only be obtained through the National Institute of Drug Abuse Drug Supply Program. NIDA's purpose is to advance science on the causes and consequences of drug use and addiction, and to apply that knowledge to improve individual and public health, meaning that research into therapeutic benefit is not part of their mission. In fact, less than 17% of the research on cannabinoids funded by NIDA in 2015 concerned the potential benefit to patients. The continued focus on drug addiction and abuse by NIDA research severely limits the potential research on the therapeutic benefits of cannabinoids. In order to receive approval for cannabis research, there are numerous hoops researchers have to jump through. First, they must submit an investigational new drug application to the FDA and DEA for review. If the researcher is planning on using research-grade cannabis, a letter of authorization must also be obtained from NIDA and submitted along with the investigational new drug application. The researcher must then apply for DEA registration and site licensure, and then wait at least 30 days for FDA to respond. If the FDA places a hold on the application, research cannot be conducted. Researchers must also apply for any necessary state licenses, such as a controlled substances certificate or approval from a research advisory panel. Researchers must also submit a research protocol to the DEA for review of security provisions concerning the storage of cannabis. The DEA requires a safe, steel cabinet or vault with limited access to authorized employees only, and researchers must bear the cost of meeting these security requirements. Finally, researchers must obtain approval from an institutional review board prior to initiating their research protocols. These many complicated steps make it very challenging and daunting for most researchers to apply for and be approved for cannabis research in the United States. As mentioned, cannabis used for research can only be provided through NIDA. The only NIDA-approved source of cannabis for research is the University of Mississippi, which has been the sole provider of cannabis plant material since 1968. The plant material obtained from the University of Mississippi may have been harvested years earlier and stored in a freezer due to production restrictions and variable supply and demand. This means the plant provided by the University of Mississippi is not reflective of the potency or various formulations of cannabis which are currently available to patients within state-regulated markets. This results in studies which lack validity and applicability to the current cannabis market. In 2016, the DEA adopted a policy which would allow for an increase in the number of private entities allowed to grow and provide research-grade cannabis. But as of the first quarter of 2020, the University of Mississippi remains the sole provider of cannabis for research. This delay in the application processing continues despite a lawsuit against the DEA by a research facility which charged DEA with intentionally delaying the approval process and essentially stifling cannabis research in the United States. Two additional complications with cannabis research are blinding and placebo control. With placebo control studies, the plant material or placebo provided by the University of Mississippi has been processed to remove any active cannabinoids, but still has terpenes and flavonoids intact. Given that these ancillary compounds in cannabis are thought to have additional clinical effect, this may not be a true placebo as provided to the patient. Blinding also presents an issue, as the intoxicating effects of cannabis make it very apparent to patients when active medication has been received, which results in an unblinding of the study and invalidates any research results obtained. Despite the many barriers to the traditional clinical research of cannabis, the number of cannabis studies has continued to increase exponentially over the past decade, as represented by this graph. Due to the lack of standard double-blind placebo-controlled clinical research studies on cannabis, the majority of the studies conducted are preclinical or observational in nature. Observational studies are inherently limited due to the lack of control and influence of confounding barriers. In observational studies, two events can be linked together, but not causally. Often in these studies, association is easily confused with causation. A classic example of association not equaling causation is that an increase in ice cream sales is correlated with an increase in drownings. Obviously, eating ice cream does not cause an individual to drown. However, both tend to occur on hot summer days, so there is a clear association between the two. This sort of evidence can easily be skewed to misrepresent the results of clinical studies, and with cannabis especially, can become difficult to interpret when we have a lack of traditional, standard, double-blind, placebo-controlled studies. There are several areas of perceived benefit when it comes to cannabis. The first is widespread therapeutic activity, particularly for patients who have failed traditional pharmaceutical therapies. This includes pain management, anxiety, PTSD, epilepsy, nausea, vomiting, autoimmune disease, neuropathy, and degenerative diseases such as Alzheimer's, Parkinson's, Huntington's, ALS, etc., among others. Cannabis is also perceived to have a low rate of addiction with no apparent risk of overdose fatality. Additionally, Cannabis has the potential to play a role in the reduction and treatment of opioid addiction. Finally, cannabis is perceived to have very mild side effects with few serious contraindications or drug interactions. The main areas of perceived concern regarding cannabis involve increased adolescent access to cannabis, an increase in motor vehicle accidents with cannabis use, the development of cannabis use disorder, reduced cognitive function or long-term brain injury associated with prolonged cannabis use, psychosis-like effects of paranoia and disorganized thinking associated with cannabis use, and the incidence of lung injury or cancer involved with smokable cannabis dosage forms. It is also important in the context of risk versus benefit to consider the adverse effects of cannabis as compared to other drug classes commonly prescribed for the same symptoms. For example, opioids have a high rate of addiction, cause nausea, constipation, and sedation, as well as severe withdrawal symptoms, and the common development of tolerance to therapeutic effects requiring continued escalating doses. Tricyclic antidepressants have anticholinergic and psychotropic side effects and interact with several different drug classes. Antiepileptics cause sedation and neuromuscular effects. Muscle relaxants and benzodiazepines cause sedation, hypotension, memory lapses, dependence, and potentially very serious withdrawal symptoms. Comparatively, cannabis causes dry mouth, sedation, dizziness, and euphoria, which is typically dose-dependent and seen with higher doses of THC.